In this video, we will discuss about this hepatitis B virus surface antigen mRNA to make cancer sensitive to the immune system. So the idea is that it is difficult to find the antigen that are commonly and uniformly expressed on the heterogeneous population of the tumors. And since it is difficult to find antigens, so any immunotherapy with a particular antigens will not be able to kill almost all of the cells. In order to take care of that problems, the idea that this paper suggested is to make the cancer cells sensitive to the microbial, make the cancer cells to express the microbial antigens, and our immune system has the efficient or enough immunogenicity against those microbial antigens. Thereby, it can trigger the immune response against the cancers through these microbial antigens that are expressed or synthetically expressed on the cancer cells. So, considering the safety, what the antigens that they have decided to express on the cancer cells is this hepatitis B virus surface antigen. The idea behind this that it will cause this hepatitis B virus surface antigen will be packaged in the lipid nanoparticles, and they will be expressed in the cancer cells using the cancer cells. And the idea is that before these lipid nanoparticles are been detected by the cancer cells, they will be expressed on the are detected by the immune system. Before these lipid nanoparticles are being detected by the immune system, they will be already expressed. They already does its function to express this hepatitis B surface antigens on the cancer cells. So the idea is shown here that the once this mRNA-H, they call it as H, is been recognized by these genetic cells, that it will cause the effector T cells to form. It will cause the effector T cells to form that can target this HBsAg HBsAg antigens or hepatitis B virus surface antigens. So first they check the lipid nanoparticle size. It is around 100 nanometer. It is appropriate. It is appropriate for this applications. And then did the in vivo studies where they inject this mRNA-H along with the control, along with this control saline and HBsCHO, and check the IFN-gamma. IFN-gamma usually produced when there is a killing of the cancer cells by the immune cells occurs, and they can see that IFN-gamma levels are high in the case of this mRNA-H. We can see it from the panel C and from the panel D also. This HBsAg peptides are high in the mRNA-H conditions. And thereafter they check this how many of these HBsAg memory T cells are formed, and they can see that this IFN-gamma and this HBsAg T cells, they checked it with the IFN-gamma, that is the T cells will secrete IFN-gamma, so they will also have this IFN-gamma plus CD8 plus T cells markers, and they are able to see that these cells are high in the case of mRNA-H compared to the other two conditions. And the tumor volumes is also suppressed in the case of their mRNA-H, and the survival rate are also high in that conditions, indicating that their system is performing better and as per the expectations. Then they check this HBs neoantigen are expressed on the tumor cells or not, and the process that they expect is shown in panel H. When they check this amount in the controls and in the this VVH, they are able to see that neoantigens can be expressed. HBsAg neoantigen can be expressed using the virus formations. And when they check it for the other control, the VVG, it can also be expressed. And thereafter they check dose dependent increase in this virus, time dependent increase in the virus amount percentage. They can see that the time dependent increase is also there, indicating that the signals for the VVH are bona fide, seems to be bona fide. Thereafter they check the specificity of this mRNAs of the HBsAg. They can see that the HBsAg mRNAs are specifically targeted to the tumors and compared to the other main body parts where there is no signals. Then they check the lysis of these tumor cells compared to the spleen cells, and with increase in the ratio of tumor cells to spleen cells, they can see that with increase in the ratio, with decrease in the ratio, that is with increase in the spleen cells, the lysis of the tumor cells are also increases, indicating that the specificities of the this mRNA and this immune systems, indicating the specificity of this immune systems against the HBsAg mRNA containing tumor cells. So this is what they have so far validated their hepatitis B virus surface antigen mRNA platform through the results, by which on one end they shows the efficiencies of the immune systems. The immune systems can be activated by this microbial antigens. At the same time, they also shown the expression capabilities of this neoantigens using the virus. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.